Alright, let's go over some dilution calculations and how to make a working concentration from a stock concentration. So the first reason that a dilution is actually quite useful is that it's actually a lot less labor to have a stock concentration and dilute that down to a working concentration, in contrast to having to make up a new solution from just new chemicals and having to weigh that out and pH it every single time. The second reason is actually if you have a stock concentration and you make that into a working concentration, you're actually going to have less chance of contaminants because there's less people going into the stock. So if the working gets contaminated, you can simply just make a new one from the stock again. So there are a few different types of dilutions that you may see written in a protocol, or just different types of dilutions that are used in a lab, and we're going to go over all of these with you. The first one is just a basic dilution where you're trying to solve for V1, which we'll define later, using the formula C1V1 equals C2V2. The second one that we see here is just some sort of number times stock, so for example, a 50 times stock or a 10 times stock. The third one would be examples of relative volume. So for example, adding 2 volumes of something to a solution to dilute that down. And the fourth one would just be a serial dilutions, which is usually used in plating of bacterial cultures to count colonies. Okay, so let's just define the variables in the C1V1 equals C2V2 formula first. So C1 is your starting concentration. This is otherwise known as your stock concentration. This is what you're going to be diluting down from. V1 is usually the variable that you're going to be solving for. It's the volume of the stock concentration that you're going to need to make your final solution. C2 is your desired end concentration. It's a lot more diluted than the C1 concentration. And V2 is just your desired end volume. So let's go into our first example of a C1V1 dilution. So we're solving for V1 here, and our example is just going to be using a 1 molar stock concentration, and we're trying to find the V1, how much we actually need of this 1 molar stock to make our 100 micromolar 500 milliliter final solution. Okay? So the first step that we actually need to do is convert everything into equivalent units. So if you noticed, 100 micromolar has to be converted into molar, and 500 mils has to be converted into liters. Alright, so now that everything is converted so it has the same units, it's just basic algebra to solve for V1, and we're going to get an answer of 0.05 liters or 50 mils. So that means that we need to take 50 mils of our concentrated stock solution and use that to make our final diluted solution and top it off with 450 mils of water. So our second example is going to be using the same C1V1 equals C2V2 formula, but we're just going to be doing it with a mass over volume type of concentration. So in this example, we have a stock of 100 milligrams per mil, and we want 100 micrograms per mil, and we want a final volume of 200 milliliters. So same thing as the first example, we need to convert so all the units are equal. So we have to convert 100 milligrams per milliliter to 100,000 micrograms per milliliter. Now the volume, the 200 milliliter volume is already in mils, so that's okay because everything else is in mils as well. Okay, so after the units are all converted, again, it's just basic algebra to solve for V1. And so we get a final V1 of 0.2 mils, which is also 200 microliters. So that's what we need to use to make our diluted solution. Okay, so we're going to go over a 6 times concentrated solution example. In this case, we're going to use the example of loading dye. A loading dye for DNA samples and running gels often comes in 6 times concentrated solutions, and you have to dilute that down with your DNA sample mixture down to 1 times. So we'll show you how to do that here. So we start with the same C1 V1 equals C2 V2 formula. C1 here is a 6x loading die, because that's how concentrated our loading die is in this example. V1 is an unknown. C2 is going to be 1 times. That's going to be how much the working concentration is. And in this example, we're going to be using a 20 microliter DNA sample mixture. And we don't know what our final volume is because we don't know how much loading die we need to add to this mixture. So it'll be 20 microliters plus V1. So now we just need to use algebra to solve for this again. Okay, so we basically just need to bring the V1s together and isolate that to one side. And we should end up with V1 equals 4 microliters. So that tells you we need to add 4 microliters of the 6x loading die to our 20 microliter DNA sample. And that'll bring that loading die concentration down to 1 times. Okay, so this is a much quicker and easier way to figure out how much loading die to add, because of the fact that this is a 1:6 dilution ratio, which is a 1 part dye to 6 part reaction or DNA sample. You're going to need to add 1 microliter for every 5 microliters of reaction or DNA sample. It's a lot easier to think about it this way than having to calculate it out using the C1 V1 equals C2 V2 formula every single time. Okay, so the next example is just using certain volumes of water or diluent to dilute a concentrated solution down. So in this example, we're using 2 volumes of water, so that means that we need to add 2 times the volume of the starting solution. So here, our starting solution is 50 microliters. So this would mean that we need to add 50 microliters times 2, so that would be 100 microliters of water that would be added. We'll have 150 microliters total, and this solution is now less concentrated than it was before. Okay, so lastly we're going to talk about serial dilutions, which is used when plating. So here we just have a diagram, and that flask is supposed to represent the culture that you're starting with. So that is the most concentrated, and usually we say or estimate that there's around 10 to the 8 cells per milliliter. And what we do is we take some of that culture and we actually dilute that further down in diluent. In this case, it would be LB. So to do a 1 in 100 dilution, what we do is we take 0.1 mils of that culture from the flask and put that into 9.9 mils of LB. So this should give us around 10 to the 6 cells per milliliter now. And we're going to do the same thing with the next test tube. So we're going to take 0.1 mils from our first test tube and put that into 9.9 mils of LB in our second test tube. So now we should have around 10 to the 4 cells per mil. And with the last dilution, what we're going to do is we're just going to do a 1 in 10 dilution. So we're going to take 0.1 mils of the culture from the second test tube, and we're going to add that to 0.9 mils of LB. And it should give us around 10 to the 3 cells per mil. Finally, we're going to take 0.1 mils of the culture from the third test tube and plate that. And that should give us around 100 cells on the plate. So if you don't know how many cells you started with, you could also just count the number of colonies that have grown on your plate and then use the dilution factor to calculate the number of cells in your flask. Alright, so thank you for watching. Make sure you hit that like button and subscribe if you enjoy this sort of content, and we'll see you in the next video next week. Thank you. Bye.